long introductions where he's read people's entire CV. He summarizes their history in ways that they didn't even know was summarizable, and I'm not capable of that. But it is a pleasure to introduce Chuck Stevens. I first met him as a student of biophysics when I was a student of biophysics, and you may have met him then too. He can only be described as an icon of that era and several eras to follow that, and I'll mention that. He was, his degrees were at Harvard, Yale, and Rockefeller, BA, MD, and PhD back in the day. Chuck was H.K. Hartline's graduate student. Who here has never heard of H.K. Hartline? Come on, raise your hand. No, raise your hand. It's okay. It's okay. We'll just say you were born in another country. H.K. Hartline won the Nobel Prize for basically describing lateral inhibition in the limulus eye. Is that right? I don't know what he was like. I don't. Maybe we'll hear about that later. I've intersected Chuck in various ways. He was a student at Rockefeller when a guy that I worked for, Jerry Edelman, was a student at Rockefeller, just a little bit ahead of him, and I circled back later. But mainly I encountered him in his papers. I moved into neurobiology in the mid-'80s, and the papers that I read were Stevens, Connors and Stevens, Magleby and Stevens, Becker and Stevens, Colacune and Stevens, et cetera. These are papers that basically define biophysics in that era. Chuck, Bertil Hille, Clay Armstrong, Pancho Gazzania, and the like of those were inheritors of Bernard Katz and Hodgkin and Huxley and Hartline. They were the next gen who defined that whole era, and we have him here to talk to us today. He didn't stop there. His work is characterized in particular by rigor and an interest in noise. Right? So when you look at brains, you get there's a lot of noise in brains. There's a lot of noise in the anatomical connection patterns of brains, and he has started to lay structure on top of that. So two of the things that have occupied him over the last 30 years, where he's been an investigator for the Howard Hughes Foundation, Howard Hughes Medical Institute, have been how does an animal optimize the wiring in its brain, and how does this represent information? How does this scale phylogenetically across species, and what are the principles by which these things have been selected for and wire up brains? I could go on and on. I'm just going to read what he's a member of. He's had the Spencer Award from Columbia University. He's a member of the National Academy of Sciences, a member of the American Academy of Arts and Sciences. He has a Gerard Prize, Ralph Gerard, I guess, from the Society for Neuroscience, the Carl Spencer Lashley Award from the American Philosophical Society, and the Stephen Clifford Lecture in 2001, just to name a handful. So it's fun to have him here and have him talk to us, and thanks for coming. I appreciate you taking the time out to be here, and I hope it didn't embarrass you with your introduction. And I apologize to the crowd for not being as eloquent and long as Mike would be. So thanks, Chuck.
Okay, thank you, Rita. It's a great pleasure to be here. Thank you for coming for my talk. Most of you are not interested in flies. I doubt if there's anybody here who works on flies or even cares about them. And the only thing that probably exceeds your disinterest in flies is on how flies smell. And this talk is going to be on how flies smell, in the sense how does their odors odors that are presented to their noses get interpreted to the brain. Now, I don't actually care much about flies either. And so the very first thing I'm going to do is take a little bit of time to tell you what the problem I think I'm working on is, that flies are the answer for. The problem, I'm going to say it in a sort of obscure way, and then I'll explain it. The problem is all animals have the problem of being able to put a neural tag on an arbitrary point in a high-dimensional space. What's an arbitrary point in a high-dimensional space, and what's a neural tag? A neural tag is a few neurons. In the case I'll tell you about, it's a couple of dozen. In your brain, it may be 1,000 or 10,000. Those neurons represent a point in high-dimensional space. I'm going to say what that is in a second. in a way that takes into account all the information that's available, and that tag, which has 1,000 neurons in it, and it overlaps as little as possible with any other possible tag for the same thing, for another point in the high dimensional space. Okay, what a point in a high dimensional space is, for example, is this is face recognition. And face recognition is getting pretty good. It's as good, the computers are about as good, last time I looked a few years ago, about as good in certain circumstances as we are. And one of the main ways of doing it is to take a face and measure off a lot of things. And so, for example, you could measure this point here, and this right in the middle of the nose, and that would be, say, P1. And then you could measure this point here, which would be the middle of the chin. We'll call that P2. And then you could make a graph of, here is P1 and here is P2, and those 2 points define a point, say, right here. A different face would have a different distance between those 2 points. You measure off all these values in some coordinate system, and so it would be in a different place, in a different point on those planes. Now, I could measure another point, say, up here. I'm going to put it right in the middle so they're in a line, and this is P3. And I can put that point on the same graph. There's P3. And then, now this point is a point in a three-dimensional space. And any different face would have the points in different places. This particular algorithm that was used for this uses about 50 points, and so every time you add a point, you add a new dimension to the space. And so basically, a high-dimensional space is, say, a 50-dimensional space, although there are higher ones, and you can characterize any face by a point in high-dimensional space. And you have to be able, the evolution can plan in how you get these points in high-dimensional space. Evolution knows all about faces. It knows you need to know about them, so it can plan in exactly how to extract the points in this high-dimensional space, but it can't possibly plan in for every possible face you might see to give you a label for it already. There's not enough space in the brain for doing that. What I'm interested in is, how do brains actually accomplish this? How do they take any arbitrary point in a high-dimensional space and put a label on it, a tag, which is a certain number of neurons that fire whenever that point is for a particular point in space, that overlap as little as possible with any other point in the space, a lot of space up there, and with any and make use of all of the information that is available to them. First of all, is it possible? Can the brain actually do that? And many of you will recognize this person. This is Jennifer Aniston, and there was an article a few years ago by Christoph Koch, who is a neurobiologist, a theorist, who used to be at Caltech and is now at the Allen Brain Institute, the head of research there, and a neurosurgeon named Fried, who was mapping out seizure loci in patients that had intractable epilepsy. And so he was going to take out the epileptic focus, but he had to make sure he knew where it was. So he took out as little as possible and made sure he got it. And in the course of this, just like Rita's been doing, they were doing experiments on the patients that volunteered for this. And one lady volunteered. There were actually a number of people who volunteered for a dozen or so who were in the paper. And they recorded from cells in the medial temporal lobe, and mostly they were some in the amygdala, but mainly they were in the hippocampus. And this cell was a cell in the hippocampus, and down here, is there a pointer? I didn't. It's not actually. I can walk over here. Doesn't matter. These are pictures of Jennifer Aniston that they showed it. If they showed it pictures of anybody else. Okay, got it. Thank you. If they showed it pictures of anybody else, the cell didn't respond, but it responded to about 10 pictures of Jennifer Aniston. These are 3 examples. This is the firing of the cell histograms. This is time here and number of spikes that came out, and they flashed these on the screen for a minute. They showed this patient hundreds of pictures. The pictures were of the Leaning Tower of Pisa, spiders, elephants, famous people. And they discovered cells that responded to each one of these, and this particular cell responded to Jennifer Aniston over and over again, but to nobody else. In fact, I'm not sure if it's true for this one, but for most of them, if you just write down on a word, on a piece of paper, Jennifer's name and show it to the person, the cell would fire spikes. So evolution could not have possibly known that you needed to recognize that face. It had to make a tag for every face that would come, and then be able to use that tag to learn a name and character properties of it. Okay, so the problem I want to know is, how does the brain manage to do that? How can it possibly take a point, an arbitrary point in a big space, and encode that point, no matter what it is, in a way that overlaps as little as possible with any other point in the space and uses all the information possible? I'd like to be able to do that for the hippocampus, and it might even be possible one day, but a place to start is a simpler system, and there are analogies, homologies between this simpler system and the hippocampus and other things. I'll mention this at the very end, that make you think that the other systems may be doing it the same way. So a place that you have to start someplace. A place to start is the easiest place you can think of to find out how this is accomplished. And so I'm going to tell you how the fruit fly accomplishes this. The fruit fly brain is a little different from the ones that you're used to. This is a section through the brain. The white things are cell bodies. The gray things are neuropil, where they're axons and dendrites. And unlike us, but like most invertebrates, all of the cell bodies are on the outside, and the gray matter is all in the inside in big lumps like this that all have names. And so, we have our cell bodies embedded in the neuropil that they serve, but the fly doesn't do that. They have it arranged this way. There is a particular, the fly system has a 3-stage architecture. The first stage here is odorant receptor neurons. I'm going to go over this a couple of times. They sense the odor. I have here 4 of them, but there are actually about 2,000 odorant receptor neurons that fall into 50 classes. Each of the 50 classes is characterized by a particular odorant receptor gene that is different from the other 49. And if you give any odor, all 50 of the odorant receptor neurons fire spikes or don't, as the case may be. There's some profile across all of them. In this case, there's only 4. This is in the nose, which I should have pointed out in the fly, but I'm not going to bother. You don't care. And then the information from these odorant receptor neurons O-R-N. These odorant receptor neurons send their axons to regions of neuropil, and I've drawn them here as little circles. And their axons end in the neuropil, and all of the cells that express the same odorant receptor gene all converge on a single region of neuropil, and so all of the information coming into this region of neuropil here is from the one particular odorant receptor protein, so they will all have the same response. These will all have the same response. These will all have the same response. Okay. Then there are this structure here that I put a box around is called the antennal lobe, AL. And the neurons that that gather the information. One neuron gathers the information from this region here. One gathers it from this region here. There are actually 50 of these, one for each odorant receptor protein. And these are called projection neurons. These projection neurons send their axons. Their dendrites are up here. They send their axons over here to another place called the mushroom body. And the mushroom body, so there are 50 projection neurons, and there are 2,000 cells called Kenyan cells in the mushroom body. So the information from these 50 neurons here, I drew 2, is spread over 50 Kenyan cells, and each Kenyan cell takes a random sample of 6 of these projection neurons. So basically, each Kenyan cell takes a sample of the information in from 6 different odorant receptor proteins, and then the output of these Kenyan cells goes out to lobes of the Kenyan cell body for learning. It's known that the fly can learn to respond to any odor you give it specifically. The flies can learn to a number of things, but mainly the experimental things are to learn to approach or avoid a specific odor. And if you give a fly an odor and then you reward it in the presence of that odor with some sugar water, the fly, when it smells that odor the next time, will approach it. If you shock the fly in the presence of that odor, the next time the odors, you can put the fly in a little tea maze with the odor on one side and not on the other. The fly will always avoid the odor. And that happens in a different part of the mushroom body down here. It's known how that happens, but I'm not going to talk about it. Okay, what I'm going to be telling you is how, if you give an odor, then you're going to have a pattern of firing, different firing rates in 50 different neurons. Every odor then is a point in a 50-dimensional space. The fly, we know from experiments, can learn to identify, it can learn any odor you give it, doesn't matter what it is. It can learn that odor and learn to respond to it. So it has to put a tag on that odor, and it's known that the tag is the firing of a certain number of out of these 2,000 cells. So we know everything about the signal that's coming in because there have been experiments, and I'll tell you more about that. And the tag has to be selected somehow by the circuitry in the mushroom body, because that's exactly what goes down goes over here to do learning on. So this fly can learn any odor. That means it can put a tag on any point in a 50-dimensional space, and that tag is unique. And so this is exactly an easier system to study than it is to study the Jennifer Aniston cells. So I'm going to be telling you how it is that the code, the combinatorial code that represents every point in 50-dimensional space is represented, is characterized, and I'm going to tell you the mushroom body. What the fly does is it looks over all the possible cells that are in there, and it picks out about 100 that are used to form the tag, and I'm going to tell you how it decides which 100 to use. Okay. This is a very casual seminar, and so if there are questions, you should stop me anytime. Just yell out and ask me the question. But let me take a second now to find out if there are questions about anything I've said so far. Is it all okay? If you think of one after a second, tell me. Okay. My contribution to this work is in two parts, and I'm going to tell you what the contribution is, and then tell you why you don't need to worry about it. Okay. The first contribution is the following. Normally, information in a region of the brain is localized. So, for example, here is the letter A, and here is the visual cortex of an animal, and there's a retinotopic map from the letter A over here onto the cortex, so that the A actually is represented by cells in a pattern A on the cortex. It's a little distorted, but that's basically it. So I can tell you, look, measure all the cells right around here, and those cells will give you all the information in the part of the visual world that's right there. The cells, the information is localized, so if I tell you a certain number of certain cells to look at, you can get information about any particular part of the visual world. That's all the information the animal has about it. This is localized information. What I'm going to claim is there are parts of the brain where information is not localized, it's distributed. And that doesn't mean it's just all over, it means something very specific. If this is a region of the brain that is getting distributed information, the information in the visual world, for example, would be spread over every place. Every cell would get some of the information from every place. It's all over the place. However, in the kind of region I'm going to be talking about, the distributed information can be gotten back from no particular cells, but from any cells at all if you get a particular number. So here are, say, 5 cells. If those 5 cells have access to all of the possible information. If I look at those, they have all the information. If I look down here at these 5 cells, those would receive exactly the same information. It might be represented differently, different weights and things, but it'd be the same information. If I look at these 3 and these 2, that would have the same information. Just matters how many you look at, not which cells you look at. Okay. Question is that possible, and how would you do it? And I'm going to tell you it is possible, and I'm going to tell you how you would do it, and then I'm going to tell you you don't need to worry about it. Okay, so my contributions were all in theory, and the theory part was to identify the existence of regions that have distributed information distributed in this particular way, and how it works, and the thing that makes you be able to get back all the information from a certain number of the 2,000 cells. There's 50 odorant receptor information types that goes to 2,000 cells. Each one of them samples at random. There's a paper from Richard Axel's laboratory which shows that each Kenyan cell takes a random sample of 5 of the 50 types of information, of 6 of the 50 types of information. And this is what's called a random projection. And there is a new theory about 10 years old called compressed sensing that's mainly due to a mathematician at UCLA called Terry Tao. And Terry Tao showed that if I give you a photograph, and that photograph has lots and lots of pixels in it, that actually, for most photographs, you don't have to keep the information from all the pixels. If you do a random projection of those pixels onto a shorter list of pixels, and you just look at the information that are in those, you can get back the photograph that you started with. This is compressed sensing, and compressed sensing implies that if you have a list of numbers. In this particular case, it would be a list of 50 firing rates coming in from the 50 odorant receptor channels, that if you spread that information at random over 2,000 neurons, that if you look at, say, 1 or 2 dozen of those neurons, you can get back all the information. And there's a theory for that, and I can show you exactly why it's true. And it's not that hard, but it takes a little bit of time. But it turns out that one of the reasons that I have been working on the fly is that the experiments that you would need to do to find out if Terry Tao was right in this case had already been done. And so all you have to do is, I'm going to show you the experiment, and then all you have to do is believe the experimental data. You don't have to worry about the theory.
Question?
Yes?
The 6 inputs per neuron. How does the cell know that? How does that know how to limit it to 6? How many inputs are on these neurons?
6.
No, just total 6, and no more than 6.
Yeah, each one gets 6 synapses total.
That's it.
Yeah.
And if they regrow, does this stuff regrow? Can you
Every fly is wired up a different way. So I don't know about regrowing, but if you start with flies,
I guess I meant, is there only room for 6?
so in the cerebellum, The granule cells in the cerebellum have mossy fibers that come to them. Each one of the cells has about 4 little arms that stick out and grab grab information from one mossy fiber, and the anatomy here is exactly the same. They're designed that way with little arms with claws on the end. Those claws grab on to one of the odorant receptor projection neurons, and they grab onto 6 of them. Okay, so this was an experiment done by Glenn Turner at Cold Spring Harbor. What he did was he trained a bunch of flies to respond to odors, and he took 2 odors, odor A and odor B, and each of the odors was neutral in the beginning. If you put the fly in a tea maze and put one odor on one side and the other odor on the other side, the fly would then would go equally. They would have a population of flies, and they would go equally between the 2 arms. Then he made compound odors that was odor A, and then 70% A, 30% B, and then 60% A, 40% B, and then 40% A, 60% B, and so forth. And he took the flies and he took them and he gave them each of those odors, and he put the mixture the mixture 60-40 on this side, 40-60 on that side of the tea maze, or 70-30, 30-70 on that side. And so if he had done 50-50, the odors would have been identical. They were barely separate if you did 60-40, and if you had 100-100, then there would be pure odor A on one side, pure odor B on the other side, and the fly would have been shocked for odor B, or odor A, or actually odor B. And if you had 70-30, 30-70, the fly would have been shocked for 70% odor A, 30% odor B, and he would be going against 30% odor A, 70% odor B. Anyway, you get the idea.
Another question.
Yeah.
Okay, this is a stupid question because I don't remember. This is Drosophila. Drosophilus, fruit flies.
Yeah.
Okay. This is shown over a population of flies because an individual fly won't show this.
No, individual flies show it, but you have to. It's quicker to run 50 flies and just count them than it is to run one fly 50 times.
That's what he would do.
Yeah, they
on the cohort, right?
Yeah, yeah, right. Okay, and the yellow line here is Glenn's behavioral data. This is the fraction of the time that the fly. This is 50% here response, and this is the fraction of the time the fly turned to the side with the shocked away from the shocked odor when it was pure odor, 70-30. 60-40, and that's here, there. If it was 50-50, the fly can barely tell them apart here, but in 50-50, the fly couldn't possibly have told it. It's the same on both sides. Okay, those are his data. Then, what Glenn did is he got a fly, a different fly, and a bunch of different flies. He revealed the mushroom body. He put calcium dye in there. He recorded the calcium signal from all the neurons in the mushroom body, all 2,000, and only a few of them fire spikes for any different odor, different ones for different odors, and about 5% fire spikes. And he did this on a bunch of animals. He collected data from about 100 different of the Kenyan cells that are getting these data, and he then did a simple linear classifier model and found that if he looked at any 25 cells, he could exactly replicate the fly's behavior. This is not even necessarily the best way of doing it, but just a simple linear classifier did this, and this by looking at any 25 of the neurons, and it didn't do any better actually if you looked at all of them. So what he has shown is that the information that the fly has available based on behavior is present in any 25 of the neurons that were firing spikes. Doesn't matter which ones you pick, and he picked different ones. Okay, now instead of hearing the theory and believing it, all you have to do is believe Glenn's data. And yes, there are 5% of the cells are firing spikes, and of that, a subset of 25 will have as much information as the fly has to give his behavior. Okay, that is how the mushroom body works. The trick is it is distributed information, so it doesn't matter which cells you pick. It means you have to pick the cells that are going to give you the best characterization of the odor. 25 cells are a tag for any odor. You give it a different odor, a different 25 cells will be its tag. Take another odor at random. So it says that all the fly has to do is look over the 2,000 Kenyan cells, find the ones that are going to be the best tag, that's going to overlap the least, and those will guarantee to be based on all the information. How does the fly look over all the possible 2,000 cells and pick the 100 of them that are going to overlap the least with anything else? In order to do that, you have to know the nature of the code that comes out of the antennal lobe. The antennal lobe has 50 channels across. Every odor makes a different pattern across that 50 neurons, and the Kenyan cells are sampling that at random. So you have to know what is the nature of the code, this 50-dimensional code, which can code millions of points in the 50-dimensional space. What is the nature of that code? Did you have a question? Yes.
Yeah, quick question. So is this a spatial temporal code like the cross-fiber patterning in the olfactory system?
No, this is just a spatial code.
Just the spatial code.
Yeah.
So the actual firing rates are not.
The rates are measured, but see the way the fly uses the information is he's trying to find a banana, say, and he flies in a sort of slightly randomish walk, sampling space, detecting an odor plume and going in that direction. And so he has to make a decision in much less than a second. And so the only thing that matters is the rate over that second. There are other animals that have complicated kinetics that are in the odor, but flies don't have that. And the complicated kinetics, we could talk about what that would be good for. But these are just the peak firing rate when you give an odor. It lasts a short period of time. Okay. What would you want the computation? Okay, the first thing is what I did is I took a random projection and I took information from an area of mathematics and computer science called compressed sensing. I derived a consequence for that, and the consequence was that if you have something that's sampling at random, you get a random distribution, and if you take a subset of that, Terry Tao promises me that will have all the information in it. That was the first part. So I drew, yeah.
Why did you go? What can you just, since it is an informal thought, what's the social story? Why did you reach for that? Compressed sensing. How did you bump into that?
Oh, I heard a talk on compressed sensing.
That's
And
why I found out about
And I thought, gee, that's interesting. I should find out about that. So I did, and at the time I found out about it, I happened to be working on the mammalian olfactory system, where it's thought that there is a random projection, but it wasn't compressing anything; it was making it bigger. I said, why in the world would you want to make it bigger? It seems like you wouldn't. The reason you want to make it bigger is you want to pick out the best ones. And you have to figure out how to pick out the best ones. Okay, what would you want the combinatorial code to be? Well, what's the purpose of it? The purpose of it is to be able to have the fly distinguish as distinctly as possibly the most odors possible. And that means you want the code that's the most informative, and according to information theory, the most informative code is what's called the charming phrase maximum entropy code. Now, if you know about maximum entropy, you love it. If you don't want to hear about it. It's a particular kind of code. A maximum entropy code is, and there's not just one maximum entropy code, there are an infinite number of them. What a maximum entropy code is it's characterized by a probability distribution, a maximum entropy probability distribution, and I'll give you 3 examples of maximum entropy probability distributions. The first one is, suppose that just 1 second. The first one is you have a range of firing rates for a particular projection neuron from 0 to 500. If you say that the probability that you take an odor at random and present it gives you an equal probability across all of those, then the maximum entropy code is a uniform distribution. If you have if you constrain the mean of the probability distribution to be a fixed number, then the maximum entropy code is an exponential distribution. If you constrain the mean and the variance to be fixed numbers, then the maximum entropy distribution is a Gaussian distribution. It turns out that Rachel Wilson's lab had shown that what the antenna lobe does is it takes all the information from the nose and it constrains the mean so that whatever odors you give, the concentration dependence is removed. And so the mean is constrained. And so your first guess would be that what the fly should do is, if you take an odor at random, then the probability for any given of the 50 channels, the probability should be exponentially distributed. In other words, you take one, I'll do that again a little bit more. How would you find this out? What you would do is you would take all 50 channels, you record from projection neurons from all 50 at once, and you'd give 1,000 odors. And then you just make a distribution of the firing rates versus the number of them there are of each one. And if it's an exponential, then that by definition is a maximum entropy code that's constrained by the mean and so forth. That's not a possible experiment to do. However, Rachel Wilson's lab did the experiment of recording from about 10% of the channels, not 50, but 7. And she said, and they didn't use 1,000 odors; they used 18. And they published a paper on this, showing that the concentration dependence was removed from the odor, so it was the pattern of firing, not anything to do with concentrations, that was determined. And they had a figure in the supplementary material that showed their data from firing rates from 7 glomeruli and 18 odors, just the raw data. And so I got my millimeter ruler out and I measured it, and it was just garbage. But I thought, well, I'll make the distribution. And so I made the probability distribution of what it came out to be, and the probability distribution is, I lost the I lost my pointer. It's okay. It doesn't. Oh, here it is. And this actually is not the probability distribution that I originally plotted. I then wrote to the guy who had done it, Vikas Bandawat, and he sent me his data. And so this is their unpublished data, and this is an exponential superimposed on it. Okay, it's not perfect. It deviates a little here and a little bit there, but there's not much data. And when you just look at it, it's garbage. It looks like it. And so I was amazed when I saw this. But what this says is it may not be exactly a maximum entropy distribution fitted by an exponential, but it can't be far off. Okay, so the first thing you have to believe is any 25 Kenyon cells is based on all the information available, and the antenna lobe encodes the odors as a maximum entropy code that gives an exponential distribution. And you don't have to, and I said, oh, it comes from probability theory, but you don't have to care about that. All you have to care about is this is the distribution, it's exponential. And it doesn't even have to be exactly exponential, it just has to be, it has to kind of go down toward the end. They didn't use many odors, and so many of the projection neurons fire faster than the 300. They go up to 500. You just have to, they didn't happen to use the right odors to do that. Okay. Yes.
So it makes intuitive sense that you want to maximize the number of odors you represent, and that's why you have this maximum entropy representation.
Yes.
On the other hand, there's pressure, evolutionary pressure. Presumably, some classes of odors are more important for the fly's survival.
Yes, right, okay, I got it. That's right. So the fruit fly is mostly, there are two parts of the fruit fly olfactory system. I talked about one part, the mushroom body. There's another part whose name is the lateral horn. These are old historical names based on how the things looked. And the lateral horn has all the information that the fly needs to behave in the world without learning anything. So there's a part of the mushroom body where all of the pheromone-related odors go. There's another part of the mushroom body where all of the fruit-related odors go. They need to find fruit. There's another part where all the odors that they need to avoid goes. Carbon dioxide is one of those. The flies fly right away from carbon dioxide. They avoid it. It's built in, and so all the things that evolution can know about is built in automatically, and all insects have the same plan for their olfactory system, and there are differences between them. Like mosquitoes respond differently than flies do because they're attracted by CO2 and not. But the point is that whatever they're doing, in order to be able to learn the best you can with what you're constrained by evolution to do, you need this process of being able to take any odor and code it as best you can to be different from any other one based on all the information, and then use it for learning. And so there are two parts. Yeah. I'm sorry, I forgot you before. Yeah.
So this neural tag of say 25 neurons, can it be expanded based on intensity? Because the functional
Odors. If you pick them at random. If you pick methanol and ethanol, you do it on purpose, then they'll fire about the same because the fly can't tell the odors apart. We can't tell them apart. They're the same odor, basically, because they're binding to the receptors the same way. Then when you go to the mushroom body, and these cells here are called Kenyon cell. After Kenyon, who described them first. Then, if you take a random sample of 6 things that are all exponentially distributed, and you say, how fast is this firing? It's possible to calculate what that is, and I can tell you what the answer is, and that is this is the probability, and this is the total rate coming in from all of the 6 samples it's done. What would that look like? It wouldn't look exactly like like with 6. It won't look exactly like a Gaussian distribution, but it's approaching a Gaussian distribution. It's not actually very different. And if you had a dozen or 2 dozen or 1,000 like we do, it would be really very close to a Gaussian distribution. And what that means is that most of the odors that you give are going to be are going to be very unlikely to make this cell fire very much, because it just wouldn't have had it just would have had 2. Oh, there we go. It just would have had too bad luck to happen in order for a cell to fire really fast. It happens to, by chance, have to sample the out of the 50 channels, it would have had to have sampled the 6 that are firing the fastest, and that's very unlikely. And so most odors will be from here over, and only a very few odors will be out there. And so what that means is that this distribution says that if I pick 2 odors at random, the chances that they will both be firing out here in the top range is really vanishingly small, because I'm having to combine 6 rare things together to get them to fire out there. And so so all the mushroom body has to do is it just has to reject all of the neurons, the Carilion cells up there, that are not firing fast or not getting a big input, and just let a few of them fire fast that are getting input. Glenn Turner has done experiments, and he shows that independent of what odor you give and independent of what concentration, always 5% of the 2,000 neurons fire spikes. The other ones don't fire spikes. Why is that? The fly has 2 neurons called APL neurons, one on each side of the brain. An APL neuron sends its dendrites down to where all of the outputs are coming. The APL neuron samples all of the information from there, and it goes back and it feeds that same information into there. It's a winner-take-all circuit. What it does is it's set so that no matter what's happening, only 5% are permitted to fire. The 5% that are permitted to fire are just the ones that happen to be lucky enough to the neurons that happen to be lucky enough, that 5%, to get the very strongest input. And because of the probabilities that are involved, the chances, if you take 2 odors at random, that their tag will overlap. That 25 neurons that you need to look at is the tag. The chances that tag is going to overlap are just nil. Okay, so that's how I claim it works. It's the simplest thing you can imagine. You just pick the fastest firing neurons and you set it all up so the fastest firing neurons are going to be the ones that are the best characterization of the odor. And if you think about it, it can't be any other way. What else? How else can you have something that picks through 2,000 neurons and finds the best one? It's just a winner-take-all circuit. It's not really an alternative. Okay, so I think it's true for okay, I'm just about done. Okay, I think it's true for fruit flies, but who cares? If you look at the structure of the mushroom body, let's see if I happen to put it here. Yes. If you happen to look at the structure of the mushroom body, it follows what is called the Marr motif. I called it the Marr motif. David Marr, almost half a century ago, made a theory about how the cerebellum works. And in David Marr's theory, these are sensory neurons. These are neurons in the pre-cerebellar nuclei. And these are granule cells in the cerebellar cortex. And what David Marr said is the sensory neurons, the first ones over there, they detect sensory information. They send it to the pre-cerebellar nuclei. The pre-cerebellar nuclei do what's called form a pattern. He didn't say what a pattern was, but there's some pattern, and I would say that pattern is a combinatorial code that's maximum entropy. Is or it isn't, I don't know. Then those cells go to the granule cells, which actually have the same anatomical structure as in the fly as in the cerebellum. They go there, and then David Marr said, you take the pattern and you separate it. He called it pattern separation. And then you take the pattern separation and you use Golgi neurons, which are very, very strongly inhibitory neurons, to wipe out the firing of most of the granule cells. And so the pattern that you use is the cells that happen to get the strongest input. Exactly what I'm saying. What I have done is taken David Marr's scheme, and I have identified the pattern. It's a combinatorial code, this maximum entropy, and I have guaranteed that the signal that you get out overlaps the least with any other possible signal, and that it's based on all of the data. Overlaps the least because it's low probability to be firing fast, and is based on all of the available information because of Terry Tao. Okay, and so the Marr motif has been around for a long time, and the Marr motif has been identified by people who work on the fruit fly and people who work on everything else, has been identified as a key part of the hippocampus, entorhinal cortex to dentate granule cells again, to the olfactory cortex, olfactory neurons, to the layer 2 input cells to the olfactory cortex, and to the cerebellum. So what I've said is this is the way it works. The trick is a maximum entropy code that makes low probability firing, high firing rate to be low probability for any particular input, and a distributed information where you're guaranteed that any output that you take that's small, that would be a good tag, is going to overlap the least possible and be based on all the information. It's possible to find that out. So the program now is to go to the cerebellum, to go to the hippocampus, to go to the olfactory cortex, and find out, is it really true that what's coming in is a maximum entropy code? Is it really true that there's a winner-take-all circuit there? Is it really true that it's a random projection or something close enough so that you can guarantee that the small sample you take is based on all the information and as good as you can get? I don't know if that's true or not, but I hope to find out. Okay, thank you.